Chapter 4 covers quite a variety of material. We'll begin with enzymes. Enzymes are proteins. Enzymes are required to catalyze reactions in the cell. That just means that they speed up reactions. The reactions would take place without enzymes, but they would be so slow that life could not exist. Anabolism is the building from smaller molecules into larger molecules. We build things to grow bodies and to repair bodies for maintenance. An example of anabolism would be anabolic steroids, where athletes use anabolic steroids to grow muscles. This reaction takes place by a procedure called dehydration synthesis. To dehydrate is to take out water, and synthesis means to make or create. Here you can see the dehydration synthesis of two monosaccharides into a disaccharide, and if you'll notice here, these two molecules are side by side, but these, look in blue, you can see two hydrogens and an oxygen. These will be taken out, and a bond will be created right here. And here in the disaccharide, you can see that this bond has been created, and here comes a water molecule. It comes out by way of dehydration synthesis. Polysaccharides are made the same way. Lipids and proteins, they're all made from dehydration synthesis. Now, fats are formed by dehydration synthesis between glycerol and 3 fatty acids to form triglycerides. A phosphoglyceride is made by 2 fatty acids and a glycerol molecule and a phosphate molecule. The bond between 2 amino acids is given a special name, even though it's still dehydration synthesis. The bond itself is called a peptide bond, and when you have 2 amino acids together, that would be a dipeptide. Many amino acids would be a polypeptide or protein. You can see here in A that we have joined 2 molecules together to form a larger molecule. This is called anabolism. This might be 2 monosaccharides to form a disaccharide, 2 amino acids to form a dipeptide. Here we've combined 4 separate monomers to form a larger piece. This might be a polypeptide or a polysaccharide. Catabolism is just the opposite. Catabolism is breaking down, whereas anabolism was building. Now, if we took out water to build, doesn't it make sense that we add water to break down? And we do. This process is called hydrolysis, which means water breakdown. We add the water to break down the molecule. Now look at these different molecules here. A, do you recognize if it is anabolism or catabolism? B, is it anabolism or catabolism? C, anabolism or catabolism? What reaction results in a polypeptide? B. What reaction represents catabolism or breakdown? C. What reaction uses water? Water is for catabolism. Hydrolysis uses water to break down. Hydrolysis, that would be C. Which reaction produces water? A and B. Enzymes control the rate that every single process takes place in the cell. They lower an energy that's required for the reaction to take place. That energy is called the activation energy. Enzymes are catalysts. They are proteins that speed up reactions. They're extremely fast. They're used over and over and over in the cell, so you don't need very many enzyme molecules. They're extremely specific. So every single reaction that takes place requires a different enzyme. So every cell needs many, many different kinds of enzymes, but very few of each kind. Now the enzyme acts by physically attaching to the substrate or the molecule it's going to change. This attachment takes place at the active site. Enzymes are proteins, and we know how important their structure is, so that active site has a very specific structure. Here you can see the enzyme in purple and the shape of that active site. We have 2 substrate molecules coming in here, for example 2 monosaccharides. The enzyme molecule stresses the bonds here, and maybe in this case it's forming a bond between these 2 molecules and forming the product molecule, which in this case, if this is 2 monosaccharides, this would be a disaccharide. We're building, or anabolism, and this takes place by dehydration synthesis, so you'd have a water molecule coming out. The enzyme is unchanged, can be used over and over again. Enzymes can be destroyed. The destruction of an enzyme is called denaturation, and we can denature enzymes with heat, chemicals, electricity, radiation. One of the reasons newborn babies cannot have a high temperature, it has to be treated immediately, is because they cannot regulate their body temperature, and at a high body temperature their enzymes would be denatured. Once that enzyme is denatured, it can never be used again.